Good morning, everybody. This is a really neat story that I love to tell because it really began organically with a phone call from the TMA about a patient who had transverse myelitis. We get these calls all the time, and I didn't know what to make of it, but I wanted to hear the story, and it started with a woman who had transverse myelitis at age 15. I'll give you more details about her case, but let me just jump to the exciting part. Her sister, 35 years later, then also developed transverse myelitis. And for a disease that's 1 in a million, to have 2 in the same family, now that piqued my interest. Because what are the chances 2 people in the same family, 2 sisters, would have the same spontaneous chance disease? So I got interested, and we got together, and I got some of the DNA, and you'll see what we did with it. But that's really how this whole story started, is just patients reaching out, offering their help, people recognizing that this is an interesting opportunity, and then you'll see the neat science that's come out of this. Now, the type of transverse myelitis that we're talking about here is the idiopathic type. Okay, this is the one that causes a single attack. It's inflammatory based. It's in the spinal cord. It causes disability at the level of the lesion and below, and then it goes away. I mean, some people heal, some people don't, but the inflammation goes away and it doesn't attack again. That's the monophasic idiopathic transverse myelitis that many of you have. And the map here comes from the TMA. Thank you, Sandy. On the website, you can find where all the members have signed up from. You can see the US and Europe are overrepresented, but the incidence is about 1 per million. And there seems to be maybe a 2-to-1 sex ratio with a bimodal age distribution. It's not really clear how bimodal it is, but we notice that there are some kids and then there's some young adults, and it seems to be those 2 patient populations. There was an interest in the seasonality. You know, everyone would say, well, I've heard, people get it in the springtime. So we looked into that. We couldn't really find a seasonality to it, but that's the basic epidemiology of TM. That's what we were jumping into with this study, and we thought, well, if there's 2 patients, 2 sisters with this disease, what do we already know about transverse myelitis genetics? And this is intended to be funny, but basically we had no idea. There were no genes involved in transverse myelitis. And it's kind of a crazy idea, right? Because if someone could be 15 years old or 50 years old before they get the disease, then what could genetics have to do with it? Because why would they go 50 years and then finally have a genetic problem? It seemed a little strange at the time, and I had the same skepticism. So here are the stories of the sisters. The first one was age 15 when she had her first symptoms. It was acute. She had sensory, motor, and bladder symptoms. These were the days before MRI. And the CSF was lost, and I confirmed it was lost because I called the hospital, and it closed, and all their records were gone. But she did respond to a course of steroids, and after about 2 years, she made a recovery back to walking. But if you ask her now, she's not doing so well anymore. But for something like 30 years, she was stable. So this is the classic picture of transverse myelitis. And when MRI finally did become available, all we could see was this small lesion here. The arrows pointing to it up here, this white line here. And if you were to cut her through the belly and look down at the spinal cord is gray, those 2 white dots. So that's the lesion, and that's what it looked like some 30 years later. So this was old damage. But it was interesting to just see that she could have transverse myelitis at age 15, and even 30 years later, you can see it on the MRI. Okay, then sister number 2, 35 years later, easily recognized the symptoms when she woke up because her sister had gone through this. Sensory, motor, bladder, but of course MRI was available. This one is an even smaller lesion because she jumped on it early, but you can see that white dot. That's it, but that caused a lot of symptoms. It did respond fairly well to steroids, and I think it's been probably 7 years now she's been stable. So this was also a truly monophasic process, not an NMO or MOG or anything like that. So we got the DNA, and there happened to be a project going on between Baylor College of Medicine, which is where Ben and I went to medical school, and the NIH and Johns Hopkins, whereby if you presented an interesting case of something familial, they will do all the sequencing for free. So we pitched this study, and they were like, wow, that is kind of interesting, we'll do that. And here are all the details about how they did it. This is really complicated stuff, lots of people involved, and it's not just the sequencing, but it's also what you do with the sequencing, because there's 3.1 billion base pairs in your DNA in every cell. That's like 3.1 billion letters in your software book about what you should turn out to be. And so once you sequence that, you have to know how to read it. If there's just one mutation, how do you pick it out among all the other genes in there and viruses and everything else that could be in there? That was the big challenge. That's what the NIH and Baylor really added to the study. And what they spit back to me about 6 months later was, they said, you know, the two sisters have an autosomal recessive mutation in a gene called VPS37A. And I was just kind of like, what's VPS? I have no idea. I've never heard of this gene in my life. And we confirmed it by PCR. And what these colors show is that right where that arrow points, the normal would be what's here in C, which is the blue. And the mutation is the one up here. And then there are people who carry the mutation, but don't have it from both parents. So these two sisters had the mutation from both parents. And they had a sibling who had the mutation from just one parent who did not have transverse myelitis. And then they had another sibling, or maybe two siblings, who were healthy and didn't have any mutations at all. Now, if you look at the general population, out of 1,000 people, one will carry this mutation. It will be just a carrier, not a transverse myelitis patient, but they're carrying the gene. So to get transverse myelitis, they would have to marry another carrier, so another 1 in 1,000 chance, and then there would be a 1 in 4 chance that the baby would have the mutation we're talking about. So it's really a 1 in 4 million chance, okay, based on the general population. So then I looked up the worldwide databases, said, well, how many people in the world have this mutation from both parents? I just want to know, and do they have transverse myelitis? So about 250 million people in the world with all of their DNA on this big database, and I looked it up, 0. Not a single other person in the world has this mutation from both parents. But 1 in 1,000 have it from one parent. So we basically had two sisters, the only ones in the world alive that we could tell, who had this mutation. So I was kind of scratching my head. I was like, I don't really know what to make of this. I'm not a geneticist, I'm a neurologist. And I honestly sat on this for about 6 months, and I didn't even know if I could publish this, who would be interested, how we could find more patients with. So then we hosted a patient day at Johns Hopkins with about 25 TM patients. And I brought up this idea. I said, can we just get some DNA from these? Just have them spit in a tube, sign a consent form, spit in a tube. I'll get their DNA. Let's just see what we see, right? We found another patient. So this next patient was also 51 when it started. She was a woman. It happened suddenly. Sensory symptoms, motor symptoms, pain. MRI was a little bit worse compared to the other patients. You could see the lesion goes, the white area goes from here to here. And if you look axially, then this whole white area part of her spinal cord. So she was more affected, but she did respond fairly well to steroids and just has pain. I don't want to say just has pain, but she's walking and has normal sensory function. So we sequenced her, and oh, by the way, she came to the patient day with her sister, which was great because I begged her for some DNA. And this is their family tree. Their parents were not alive, but the circle here is the patient, and her sister was a carrier. This is the first family I was talking about with the two sisters and the healthy sibling, and I think, I apologize, there should be a fifth sibling here who was healthy without the mutation. So then I started getting interested in this gene. Well, what is this gene? VPS37A. I had to memorize it and look it up. Basically what it does is every cell in the body has this protein, VPS37A, and it's responsible for helping to pull in things from the membrane, proteins from the membrane, and the cell is going to interrogate it and decide whether or not it's worthy to go back out and do its job, or does it just need to get recycled. Now, this machinery includes over 30 proteins, and they're all listed here, and they're divided up by all kinds of classes. And VPS37A is right here. It's just one of many proteins involved in this system. And as it turns out, it's expressed everywhere. The picture on the left is the gut. Picture on the right is a tumor cell. Okay, so it's basically involved. Every cell in the body requires this sort of interrogation of your proteins to make sure that your proteins are healthy and they should be on the membrane. Sorry, there's one more thing I wanted to show. Okay, yeah, this is it. It just so happens that there are, among all of those 30 proteins, there are other diseases that have mutations in these proteins, and most of them are neurological. So again, these proteins function in every cell in your body, but if you have a mutation in them, you can get Alzheimer's disease, a major risk factor for Alzheimer's if you have a mutation in that one called TSG101. If you have a mutation in CHIMP2B, you can get Parkinson's, ALS, frontotemporal dementia, depending on which mutation you have. And then we got an interesting report of a family born in the Palestinian territories with 6 kids, half of whom had this spinal cord. They were born with a spinal cord, what's called a hereditary spastic paraplegia, and they had a different mutation in the same gene that I'm talking about, VPS37A. So it's a different mutation from the one that these transverse myelitis patients had, and they were born with persistent spinal cord problems. So this became really interesting to me, and as it turns out now, there are several genes that predispose you to an autoimmune process. And the thought is that a lot of these genes interact with viruses and environmental triggers, and it's how you respond to that environmental trigger that determines whether or not you're predisposed to get an autoimmune disease. For example, there is a mutation in a gene called TLR3. This is a popular thing to study now because people who have a certain mutation in that gene, when they encounter the herpes simplex virus, they get an encephalitis. But if you don't have that mutation and you encounter the same virus, you don't get the encephalitis. So why do some people get encephalitis when they get a viral infection and some don't? It's your genetic makeup. And we're only beginning to scratch the surface of which genes are responsible. And this is an early list. Now we can add VPS37A to this, but we need to understand how these all work together. So to do that, one thing that I want to mention as well is the Transversemyelitis Association gave us a little bit of money to sequence the rest of this gene. So not just the ones where we found the mutation in the sisters, but what about the rest of the gene? Are there more mutations? And indeed there are. We found 8 patients now who have mutations in this same gene, but in a different part. So exon 6 was the original one where we found the 2 sisters and that one lady from patient A. Then we started screening patients using samples from Accelerated Cure Project. So if you ever donated your sample to the Accelerated Cure Project, thank you, because we downloaded those samples and all the data and found additional mutations, one in exon 7 and one in exon 5. The one in exon 7 has never been reported ever in the world, and yet we have 2 patients with that. So that's kind of unusual. And with exon 5, again, there are people walking around as carriers, but nobody who actually has the double mutation. So we think this is specific to transverse myelitis. And to prove that, what we did is we created a mouse. And this mouse has a way that we can feed it a certain hormone, and when it gets that hormone, the gene pops out of every cell in its body. And so what we're hoping to do is breed these mice up, pop the gene out, and then just see what happens. First, in a sterile environment, do they develop any spinal cord dysfunction? Is it really necessary just for normal spinal cord function to have this gene? And if not, let's say they're fine, then we expose them to infections. And I'm not sure which ones yet. We're going to get some infectious disease consultation, figure out which infections really predispose to transverse myelitis, and maybe try those. And then if that doesn't work, we're also going to try to induce an autoimmune process and see if they're more susceptible to spinal cord localization with that autoimmune process. So we have these mice, they're now breeding up, and pretty soon we'll be popping out that gene and reporting back if Sandy invites me back next year, Sandy and Chitra. So you've seen my kids. Thank you very much for your time. Happy to take questions. Yes.
I know you're a neurologist and haven't had much to do with genes, but just listening to you here, I have 8 brothers and sisters. I have 30 cousins on my mother's side, 25 on my dad's. That's 55 first cousins, 8 siblings, and here I am, 1 in a million. Which has really got my attention because there's got to be a lot of carriers in my family, or you would think 1 in 4 is a carrier. Plus, I have 2 other autoimmune diseases, one which is caused by a gene called the JAK2 gene, which is my bone marrow, which is a mutation that causes my bone marrow to produce too many blood cells. So I don't know. I want to follow you around.
Isn't that interesting? So I bet there are a number of genes that predispose to autoimmune disease, and most of them are probably involved in your interaction with the environment. JAK2 is probably on that list. I bet the list is going to be a lot longer as we get more siblings and more patients with autoimmune disease sequenced. These sequences cost about $1,000 a patient. That's all it is. But then it's the processing and the filtering that really takes a lot of time. So I think in the UK, for example, they have studies now in NMO with 2,500 samples. And they're sequencing all of them. And we'll be able to download that and start filtering through it.
And my other question is, what have I passed on to my children? I also am the only one of 55 first cousins who has twins that comes down from the mother's side, paternal twins.
Yeah.
So again,
we think these are genetic predispositions, which does not necessarily mean you're going to have the disease. If say that the sisters that we did sequence, if the one who was 50 when she started, if we'd gotten her DNA before she turned 50, what would we have told her? We'd be like, well, you have the mutation, but you're healthy, right? And then, lo and behold, she gets the myelitis. So we don't even know, and we don't know why that happened. Was she exposed to some bug or not? We just have a lot more questions than answers at this point with the genetics.
I know you're not going to like this question, but my wife, and it's about the flu shot. I know everybody dodges it. We were here 2 years ago when Dr. Carlos said that they couldn't find anything. The same days, 5 other people came up to us in the same year she had a flu shot and said that they got the symptoms. 2 weeks before the flu shot, she was at the Cleveland Clinic with all types of blood tests and passed perfectly good health. Now this year we come, there's 3 or 4 other people that had a flu shot, but nobody ever talks. There's got to be some kind of going on here out of 200 and some people that they're here.
Yeah, I think doing a lot of the vaccine work in animals.
Thank you.
I think that any vaccine activates the immune system. So if there was a process in place and all it needed was one little final straw that broke the camel's back, that vaccine is going to take it up there. That could have been an infection, it could have been a vaccine, it could have been whatever it is that's going to activate the immune system and allow that rogue element to then go attack the spinal cord or attack the optic nerve. I think vaccines, just like anything that activates the immune system, can break that threshold and allow the immune system to do its damage.
Could you do like a DNA from those people that maybe helps you guys
Yeah.
get a line that would these people not maybe get it?
That would be an interesting study because, there is a vaccine compensation injury board, and if they allowed us samples of everyone who's had an adverse reaction to a vaccine, maybe there's a genetic predisposition. Absolutely. That would be a pretty big project, but yeah. That's the kind of thinking, though.
I have a question about your dormant. I don't understand what a mutant gene is, but would it be probable that my son or my family just laying dormant because of me that I could possibly pass that gene DNA genetically down the pathway, and it's laying there dormant? Is that kind of how it works?
Yeah, so basically if let's say you're going to pass that on to a child, then that child would also have to get that same mutation from his mom. And so that's a 1 in 1,000 chance that you married that person and a 1 in 4 chance that she passed that mutation on. So any TM patient here would have a 1 in 4,000 chance of having a baby with TM if they have that mutation, just by virtue of, the prevalence of this mutation in the general population. There are hot spots. There are populations which have a little bit more or a little bit less of that mutation, but generally Caucasian populations, about 1 in 1,000.
So if we were to draw blood from both my daughter and my son, and is there a way or a blood test to show that DNA? Or just
Yeah.
DNA?
Oh yeah, we've been asking for people to send in their DNA. We only screen family members if you are positive, because there's no point in checking your family members if you're negative. Yeah. Yep. And then here.
I was just wondering if, okay, you get the information, you know it's like a genetic component. What can you do with it? I mean, can that benefit someone? Like, can you now manipulate the gene so that you can fix it?
Not at the moment. This does happen to be amenable to that CRISPR stuff you've been reading in the news because it's a single mutation, but that's not even ready for prime time. We're talking years down the road. Yeah, maybe it could be fixed, but the first thing I have to prove is that mutation is definitely going to cause transverse myelitis in you, because why put you through all that if it ends up not really being intimately linked? But so far, all the data that we have says that if you have that mutation in this world, you already have transverse myelitis. So, but we haven't screened enough patients to make any kind of claim. We're wrapping up. Okay. Thank you, everybody.